Perfect. Yeah, no problem. We'll go ahead and start. So we're going to split this talk up. I'm going to take the first half, and Emily will take the second half. Good morning from Pittsburgh. I'm enjoying my coffee here at 10 a.m. on the East Coast. Thank you for the invitation to present today at the Transplant Child webinar. We here at the STARZL Network, which you'll hear more about, have really enjoyed learning all of the work that you guys are doing, and the collaboration has, I think, been very fruitful for us. At least I can say that. So we've been asked to give a talk here. Again, you see the title about the development of multi-center consensus treatment plans for immunosuppression after pediatric liver transplant, reducing variability, creating comparative efficacy opportunities. Let's see, we'll advance the slide. So as it relates to this talk, we've got nothing to disclose, neither Emily or I. So objectives today, we're going to do a little introduction into the kind of concepts of the learning health system, a little 101. Introduce the STARZL Network as a model learning health system focused on pediatric transplantation, and then spend about the second half of the talk talking about our work developing what we are calling consensus care plans, and then how we are leveraging those care plans to perform comparative effectiveness trials here in the United States. So first of all, Learning Health Systems 101. Again, this isn't a surprise, I think, to most folks on this call. This is just kind of a pictorial of the traditional way in which research impacts our patients at the bedside. Starting with the basic research, moving its way through translational clinical trials, and then ultimately adaptation and dissemination out into the communities. The problem is that historically, I think most folks can agree this has really kind of failed to deliver the best care, or at least the most timely care for individual patients. And probably one of the biggest barriers to doing that is this concept of how long it takes for some of these discoveries to actually reach the bedside. And so this is just an article from a few years ago from JAMA, which noted that it takes on average 17 years for evidence to change practice. And again, this article was kind of focused mostly on implementation science, but again, I think QI, implementation science, all of these things are looked to be kind of adapted and addressed through the Learning Health Network structure. I think another really important gap in how we provide the best care to our patients is really kind of the lack of patient involvement in setting the research agenda and setting priorities for what is important to them as it relates to the work that we do. And so, here we're kind of talking about the concept of what we call patient-centered outcomes research. And this is kind of a very well-described subtype of research, right? And specifically, it is research that answers questions that are critically important to patients themselves. But in order to answer that, you have to ask them what is important to them. And again, we kind of will talk about some of the engagement issues as we move forward. Because what we need to do is actually then, as noted, kind of engage patients, but also all other stakeholders, right? This kind of even extends beyond the patients themselves to anybody who's involved in the process, whether it's regulators, policymakers, et cetera. And we have to engage them throughout the entire process. Ultimately, what this work looks to do is again, perform what we call comparative effectiveness trials, right? So recognizing that there's variability in practice, how can we truly determine whether option A is any better than option B? It wants to produce evidence that can be applied in real-world settings, and it helps patients and clinicians really make informed healthcare decisions about their care moving forward. In addition to this process for research that takes too long and the absence of patient involvement, there is an additional list of things that can complicate doing this in practice, right? And as it relates to transplants, I think those include outcome variation. Those include lack of clinical and surgical standardization. That can include deficiency in transparency, right? I think a lot of centers kind of keep their experiences close to the vest. And that prohibits, obviously, collection and analysis of quality data. And when there is new technology, I think many centers are often too slow to actually leverage that technology to impact their individual patients. So how does a learning health system address these challenges? What is it unique about a learning health system that we use to overcome some of these barriers? So learning health systems, were initially described by the Institute of Medicine in 2006, and they were intended to enable, again, continuous improvement and innovation, with best practices seamlessly embedded in the delivery process and new knowledge captured as an integral byproduct of the delivery experience. They aim to integrate research, improvement, management, and patient care such that every patient receives the right care at the right time, every time. And success is really dependent upon involvement of all the stakeholders that utilize and interact with the system. And again, this extends well beyond the patients themselves and includes providers, administrators, policymakers, industry folks. It's really a broad net of involvement of the folks that need to be involved when developing one of these systems. So as you construct and maintain a learning health system, what hopes to happen over time is that trust emerges as relationships among sites and stakeholders develop. Variation in care is called out, it's recognized, it's reviewed, and then it's considered within the system. And ultimately, opportunities to learn from each other and improve are identified and then acted upon. And so although this kind of structure kind of lacks the kind of rigor and formality of a kind of placebo-controlled blinded clinical trial, ultimately the hope is that change can actually be accelerated, right? That crowdsourcing can be leveraged and that quality improvement methods can be utilized to translate evidence into practice much more efficiently than has traditionally been done. And so under ideal circumstances in a learning health system, you learn from every patient that is treated. So every patient becomes an opportunity to learn and to advance. So, how does the STARZL Network, kind of embody these concepts of a learning health system? Again, we have this traditional schematic of a healthcare system where, as you transition from science to care, there's fallout, there's opportunities that are missed. The patients and the clinicians are kind of operating in silos independently of each other, and ultimately the patient experience is not ideal. And what you want to do in these learning health systems is really kind of change the culture such that all of these things are much more integrated into the care and much more aligned, such that improvements can be made much more seamlessly. And so we founded the STARZL Network in 2017. You here see our vision and mission statement. The vision was a network of pediatric liver transplant institutions committed to continuous improvement until every child can achieve a long and healthy life. And we had a mission statement, which was to unite big data, technology, patient advocacy, and transplant thought leaders to deliver the best possible care and develop new, scalable solutions to pediatric transplantation's most challenging problems. And so in 2018, we had our first inaugural meeting. We had 10 initial centers that were part of this meeting. And we, kind of wrote this publication up that folks are welcome to go dig into. But I kind of use this as a starting point for how we got to where we are. And we actually started with some very simple concepts, right? Now, again, everybody in the room, we had patients, we had policymakers, we had advisors, we had technology folks, we had industry folks, and we asked a very simple question, right? Which was, as you think about the transplant journey, what problem would you like to solve? And we had everybody just, just kind of, write, the various things that they wanted solved over the course of the transplant journey. And we had broken it up into kind of pre-transplant, peri-transplant, early post, and late post. And everyone just went around. We had whiteboards and pieces of paper, and they just kind of wrote down what they wanted the biggest problem to solve. And so again, this is kind of a lot of text, but once we had all of that, we went back and we kind of tried to look at what everyone wrote down. Again, it's spread out throughout the arc of the transplant journey. And we tried to kind of cluster these things into what were some common themes that were developing from folks as they thought about problems to solve along the transplant journey. Ultimately, what we did with that information is we kind of developed, kind of within these larger themes, we saw, areas of transition of care. We saw surgical practice. We saw immunosuppression. We saw, kind of patient-reported outcomes. We saw, kind of broad themes developing. And so we grouped them into kind of these 6 subcategories. And then we said, okay, well then, how can we still figure out where we want to start to kind of move the needle forward, recognizing that there are a myriad of issues within transplant that I think one could think to address first. But we did here these spider graphs, and again, I apologize if you can't read the font, but we kind of we plotted it out, and the kind of going around the target there, we had participant prioritization, we had size of the impact, level of innovation, ease of the innovation, timeline, whether external expertise was needed, and then project costs. And so you could see kind of objectively how we thought to kind of help us determine where we wanted to start first. Kind of based on these metrics. And so ultimately what came out of this was kind of our 4 legacy projects within the network, which is immunosuppression optimization, which is perioperative practices, transition of care, and quality of life. And so these have been the 4 projects that the network has been historically focused on, and we can talk about some of the substructure as we do some of this, and we'll use the immunosuppression working group as an example when Emily talks about the consensus care plan development. And so since 2017, we've actually had tremendous growth within the network. We've grown in the number of centers. Again, you see there in 2017 on the far left, there was only 10 centers. We grew to 16 centers in 2022. As of 2026, we are now up to 26 centers. You can see that we leveraged what had been initially a very generous donation, a philanthropic donation that helped get the network started, but we used that, those funds to help, leverage what would be needed to be built within the network to be successful. One of which is our STARZL Network data mart, which we won't talk about in this talk, but happy to kind of explore with other folks. What we did there was we kind of recognizing that there are reporting data that needs to go into UNOS and other regulatory bodies. We figured instead of just having another kind of data registry, we would partner with those folks, and then anything we wanted layered on top of the regulatory data, we did so within the network. And so we launched the data mart. We also had found early success in securing external funding, which again leveraged some of those early philanthropic gifts to kind of keep the momentum going. And so we've been kind of very successful in growing the network. And so as of today, by the numbers, we've secured over $2.5 million in external grants, and these have come from a variety of different sources, including the AHRQ, the PCORI Institute, which is the Patient-Centered Outcomes Research Institute, as well as some local and kind of regional opportunities. You see that we have over 8,000 active patients within our data mart, which again is linked to the UNOS registry here in the United States. We've had 15 peer-reviewed publications. The network itself accounts for over 65% of transplants done by active centers. We've worked very hard to engage those patients that we noted was part of the goal up front, where we have over 50 committed, engaged patient and family representatives that are embedded in every aspect of the network at all leadership levels and in all working groups. And again, we have over 26 active centers currently, including international sites in Canada and in Japan. Again, just a little bit more by the numbers. You can see here, again, broken down by center, the number of transplants that are done within the STARZL network in the last year, 2025. 68% of transplants done in the United States were done at STARZL network sites. So again, a little background of the network itself, right? And so now we want to kind of shift gears and turn it over to Emily, who will talk about, kind of how we've leveraged this kind of this infrastructure of a learning health network to develop the consensus care plans and then use those care plans for opportunities for comparative effectiveness trials.
Emily? Great. So as Jim mentioned, in the STARZL network, we picked 4 legacy or priority projects, and we're going to focus on the optimizing immunosuppression part of this. We really wanted to think together, we recognize that everybody uses immunosuppression, that we use the same handful of medications, but that everybody was doing it a little bit differently. And we hadn't taken a lot of opportunities to really share the details of our practice and think about how we could get better at this together. So we set up the immunosuppression working group with the goal of optimizing immunosuppression following pediatric liver transplantation to try to find the balance between enough immunosuppression to protect the graft, but limit the burden placed on the child and family. As you all well know, this is a sort of central issue that we're all dealing with for the child and the family, taking care of them for a lifetime after transplant. So why immunosuppression optimization? As you know, pediatric recipients live decades following transplantation. So this constant balance from the beginning of immunosuppression really has a strong impact on both their immediate quality of life and outcomes, as well as their very long-term outcomes. We know that supporting long-term graft health through sustained immunosuppression and limiting rejection episodes decreases the need for retransplantation. But again, if we over immunosuppress, then related comorbidities will increase with time post-transplant. And, you could argue in the early days of transplant for pediatric liver, at least, that this was almost worse. You accumulate, as you know, kidney problems, potentially diabetes, and increase the risk of cancer. We tried to see if we could find some evidence-based answers and reach a consensus based on current literature, but what we really found was contradictory results, and we'll show a little bit of that from looking at least the US-based registries. And we know that there's no evidence-based guidelines for immunosuppression following pediatric liver transplantation. There's not really what sort of generally what people do and that's accepted, but there's not a really rigorously developed best practices that, at least in the US, that we've studied across centers to really figure out what's the best option for a particular child at a particular time. So when we were thinking about how to right-size immunosuppression, as one often does with learning health networks, we thought about the sharing seamlessly and stealing shamelessly idea. And so we looked to other, particularly pediatric models of networks who work on rare disease to think about how have other folks aligned this, and could we sort of adapt those principles to what we were hoping to do. And so we actually modeled our development of the consensus care plans after CARA, the Childhood Arthritis and Rheumatology Research Alliance. They'd spent a bit more than a decade going through a similar process of developing standardized treatment pathways that can be developed initially through literature review or a sort of systematic, rigorous consensus process, and is really intended to reflect the most commonly used treatment approaches. They're meant to provide a more limited menu of options to choose from that reflect the current standard of care. So they're not necessarily meant to say everybody has to do it the same way, but can we provide a sort of more limited number of options that you're comfortable with choosing one of for a particular patient that might then allow us to compare more accurately across centers and do what we call comparative effectiveness research. Consensus plans, again, are not meant to dictate which medication should be used for an individual patient, but if you're going to use this medicine, here's one of the 3 dosing options that you agree to use. It's still very intended to leave flexibility to individualize care plans to the patient. It's also not meant to dictate changes to the treatment plan. It's meant to sort of provide you a starting point and potentially some next steps. But again, I think there's the acknowledgement that this is sort of clinical care that's complex and changing, and so not meant to dictate what you do, particularly over time. Here's what this looks like sort of in visual form. On the left side of your screen, you can see an example from CARA. The top pie charts show the methotrexate and glucocorticoid doses that everybody reported starting with. As you can see, there's lots of different pie pieces that are relatively small. So people were using the same medications, but in a huge variability of dosing. And through a consensus process, where they kind of thought about their current practices and worked together on narrowing down those options, they managed to come to 1 methotrexate dose that people agreed to start with, and then 3 different options for corticosteroids. And then as you can see on the bottom, there are 3 different ways that they combined these standardized doses. Again, to give people flexibility in their treatment, but also to narrow variability so that the methotrexate only that you're getting at one center is the same dosing that you're getting at another center, allowing you to compare more effectively across centers. On the right, you can see data from immunosuppression immediately after transplant. Each thin bar represents a percentage of the one center in the US. This is from the OPTN data. And as you can see, each bar is mostly one color. That means each center uses mostly one option for their patients. Our immunosuppression practices are much more center-based than they are individual patient-based. I should note that this is immunosuppression immediately after transplant. So we thought that this might reflect an opportunity where there is variability, but each center does the same thing most of the time for their patients. So we might be able to come to some consensus or narrow down the options for this variability. So why induction immunosuppression? We knew that centers write down their protocols for this. So one way that we could start in terms of transparency was just to have their centers all upload the written protocols. This gave us like a starting point so that we could look at least what people reported they were doing. We know there's significant variability, but as I kind of showed, and as you guys know, there's a standard set of medications that are used. So we're not dealing with endless variability. And as you can see on the right, the types of medications, at least in the US, that people have been using is relatively stable over time. So centers may adjust, or one center may change if they're using ATG or they're using basiliximab. But actually, if you look at the percentage of pediatric liver transplant patients that receive each different kind of induction therapy, it's been pretty standard over the last many years. Next slide. Here's what our initial look at variability within the STARZL network looks like. So we just lined up the centers and we kind of summarized what they do. Here you can see, going from left to right, the steroid dosing, the goal for coming off steroids, whether they use mycophenolate, whether they use antibodies, ATG or basiliximab, and then initial tacrolimus dosing and goal tacrolimus troughs. And so we lined up and tried to kind of categorize the dosing to figure out if we could narrow down this variability as a first step. And this was all extracted from their written protocols. This shows the process that we did after that. So again, we identified immunosuppression immediately after transplant as our initial target for developing consensus care plans. We collated the protocols from across centers and created that medication-specific protocol summary. Then for each of the medications, we had a meeting to review the current sort of evidence around that medication, as well as show the variability in practice. And then we met together to create a draft of the consensus care plans once everybody had had a chance to think through them. And this was representatives from almost all of the different STARZL centers. We really wanted to make this a very transparent and iterative process so that people would feel like they had a chance to weigh in and to think about whether this would work for their centers. After that, once, again, this is a couple years into the process already, we distributed the draft of the consensus care plans, which we'll show in a minute, to all the centers. And Jim and I actually went around and met with each center virtually and really asked them to invite all of their immunosuppression decision makers, not just the representative that had been on our meetings, to think about this. And we incorporated additional feedback, made some additional revisions to really try to make sure that this covered the current spectrum of immunosuppression and was going to be usable for people. We then sent the consensus care plans to all 16 centers at the time and asked for institutional sign-off to achieve sort of a formal final consensus. This is what our consensus care plans look like. As you can see, we have consensus care plans for basiliximab, ATG, mycophenolate, steroids. And then actually what we found for tacrolimus was that there was somewhat less variability than we thought. That for months post-transplant, at least people's sort of starting or general goals were relatively aligned across centers. It was interesting. There were actually a couple centers who, when we lined up the written protocols, realized that their troughs were much different. They were really outliers. And that actually led to some sort of initial even regressing towards the mean, where centers realized that we're doing something really different than other centers. Let's kind of reconsider that. So there was a little bit of alignment in these goal tacrolimus troughs. Again, recognizing that you're going to sort of adjust this on an individual basis for each child, but at least generally the window you're looking for was pretty common across centers, at least in the year post-transplant. Next slide. We'll just walk you through what these consensus care plans looked like. Here you can see basiliximab. Actually, every center but one used weight-based dosing. This is like sort of standard on the package for this, where the dose is given on post-op day 0 and 4 and depends on the weight of the patient. There was one center, actually Toronto, uses more specifically like mgs per kg dosing. And so again, interestingly, this was kind of an impetus for them to look at their data and look at the dosing that they gave and the outcomes. And they're hoping to compare it to outcomes for folks who are using this more sort of standardized dosing to look at the impact of that specialized mgs per kg dosing protocol that they're using. But since there was consensus amongst all of the other centers how they were using it, this is how we chose our consensus care plan for this one. Next slide. This looks at thymoglobulin. Again, some centers use this for most patients. Some centers never use this immediately after transplant. But amongst the centers that do use it, we recognize kind of two patterns. We call this standardized, where people tend to give about 5 mg per kg in 1 to 2 doses on the first 2 days, and then are not giving any after that. Versus other centers that use what we labeled a monitored kind of dosing, where they're giving 1 to 2 mg per kg per day, usually over a longer period of days, and often up to a higher max. And they're monitoring either lymphocyte or CD3 counts to try to help them decide whether to keep giving doses or not. So again, you'll see this doesn't necessarily even dictate the exact dose you're giving. Even within these categories, there is some flexibility, but it creates some categories that at least help us, what we felt like in a clinically meaningful way, categorized kids so that we could look at these two different approaches to dosing thymo between centers. Next slide. As you can imagine, some of the medications were a little bit more complicated. So again, some centers use MMF, mycophenolate mofetil, CellCept, Myfortic for almost all their patients immediately after transplant. Some centers use it for a specific subset like autoimmune or retransplant, and some centers never use it. So again, we didn't say that you had to use it or not, but we created some dosing categories, low, medium, and high, where people start the dose. And then we asked people also to try to categorize a length of treatment. How long do you intend to keep the patient on MMF? These consensus care plans are meant to be sort of written out what your intention is at the time of transplant, or actually within 48 hours afterwards. But, you know, with these that go, and that's probably pretty accurate, right, for the antibodies that you're finishing within 4 or 7 days. But for something like this, our goal is to capture the intended, and then later to capture the actual, which again will serve its own purpose in reducing variability, I think, because we'll be enable people to look at, okay, here's what I said I was going to do. Here's what we actually did. How well does that line up across patients? And I think that's will be over time another opportunity to potentially narrow down these options as we understand what actual practice patterns are. Finally, corticosteroids. This was, again, as you can probably imagine, the most variability and the most complicated. And so we spent a long time trying to figure out, and this was, I think, the most iterative process in coming up with different options. Here's what we decided. We decided to ask people to record, again, intended and then actual dosing for the corticosteroid pulse given on post-op day 0+1. We found, again, that some centers gave almost everything on post-op day 0, some on post-op day 1. But generally there was like the biggest pulse given in the first 2 days. And we felt that clinically it was made sense to sort of capture that big pulse. We then asked folks to, again, give intended versus actual dosing on post-op day 7, post-op day 30, and what their intention was for having patients off of steroids. Again, you can see within each of these categories, there's still a range of dosing, but we felt that it kind of on each time point captured low, medium, and high. And we did not attempt to dictate if you start in the low dosing, you stay there, or if you start in the medium dosing, that you stay sort of flat across the row. The intention is that you pick one at each time point, and you can adjust based on what your center's practice is and what you think is right for this child. But even though this is still a lot of options, it will, we hope enable us to sort of trace some paths and understand what people are most commonly doing so that over time we can limit down the options. Once we developed these, starting from a goal of optimizing immunosuppression through this formal plan of trying to reach consensus, then right now what we're working on is implementation and working towards comparative effectiveness studies. So now that we have these documents, people have agreed they would like to use them. We're working on actually helping centers set up implementation plans. So we developed some tools that go in the electronic medical record, or where you can fill out a quick survey within 48 hours of transplant to record what you're actually doing. And we're doing some implementation, more like QI cycles, to help centers work this into their normal workflow. What we're really hoping to use this for, and we've applied for funding and are waiting to hear back from it, is comparative effectiveness research. And the trial that we've proposed is called Optimizing Immunosuppression After Pediatric Liver Transplant. So just as a sort of quick reminder, comparative effectiveness research is a particular kind of research where you're comparing options that are already used in practice, or where there may be trials that show that either of them work, but there's no head-to-head comparison. Comparative effectiveness trials are not meant to develop new medications, but again, meant to compare options that are already in practice. So we felt like this was a really great option for us because we know from the registry data that people are using these different options. And but we also have never compared them against each other. We don't really know what works best. So here's what our proposal for the optimal trial looks like. It's a prospective observational trial. We'd like to enroll 500 children 0 to 18 years of age undergoing their first liver transplant at a STARZL Network center. So in the trial as currently proposed, that would be, I think, 20 centers, sorry. Here are the aims: to compare the effectiveness of using MMF versus not. For achieving what we call the CTPO, it's a composite transplant primary outcome at 1 year post-transplant, and also for reducing the healthcare burden on the child and the family during the first year post-transplant. And we've defined that burden as how many days you're hospitalized and what your tacrolimus and corticosteroid exposure are. Certainly, there's lots of different ways to define healthcare burden, but and to define sort of what you're trying to achieve post-transplant. But these outcomes in particular were developed through a collaborative effort of our providers and actually the families that we take care of, at least for the funding agency that we applied to, which is called the Patient-Centered Outcomes Research Institute. That patient-centeredness that you're really studying in comparative effectiveness, something that matters to patients and families, and that is going to make a difference for how they can help make decisions about their healthcare, or how their providers make decisions about their healthcare, is really important. So the first comparative is comparison is MMF versus no MMF. The second comparison is antibody induction, which could be either basiliximab or ATG, versus steroids only on achieving this composite outcome plus the healthcare burden. And then the third impact is to look at the third aim is to look at the impact of MMF and antibody induction on the child and family burden and quality of life in the first year after transplant. So these are more patient-reported outcomes, as well as some more specific immunosuppression-related complications requiring hospitalization. Next slide. Here's a sort of schematic of the hypothesis underlying the trial. Again, as you guys know, we took our consensus care plans, and we know that centers will use slightly different categories or different decisions based on some patient-specific factors like their underlying liver disease or pre-existing comorbidities to decide what their post-transplant immunosuppression is going to be. We know that too little immunosuppression can lead to rejection, and too much immunosuppression can lead to infection and other toxicities. Plus, the sort of burden of care we spend monitoring the patients for all of these pluses and minuses of immunosuppression. And it's components of this that go into our composite transplant primary outcome, which is really, again, meant to try to reflect that balance between too much and too little immunosuppression, recognizing that if you go too far either direction on the spectrum, that's not a good experience for the child and their family. Here's what the definition of the composite transplant primary outcome looks like. It balances components of the health of the liver with the health of the child. You can see the different components that we chose here. And this, certainly doesn't capture everything that's important after transplant, but the attempt is to really capture things that are immunosuppression related, and that we could, directly connect back potentially to what we do with immunosuppression immediately after transplant. We'll be looking at 6 months, and then a year will be the primary outcome, hopefully, for this trial. Next slide. So in conclusion, what we think is that learning health networks really represent novel systems of change with the potential to more urgently address challenges in medicine. The STARZL Network we've been really excited about is a novel collaboration focused on bringing the learning health network culture to pediatric liver transplants. And we've really, I think, benefited from learning from how some of our other colleagues in other pediatric rare diseases have worked on this and been able to adapt those models to the work that we do. And the consensus care plan development represents a novel collaborative effort to advance the field of pediatric liver transplantation, which could be applied to many facets of pediatric liver transplant care. So actually, we've had a couple other folks within the Optimizing Immunosuppression group, as they've seen this process go through, think about, maybe we should try, thinking about this, recognizing that it takes a long time to sort of think through for something like T-cell mediated rejection or even antibody mediated rejection. And we recognize that those, even those two different kinds of rejection, the centers tend to be at very different places in terms of how established their protocols are, how sure they are what to do. So the idea is to kind of adapt the plan development to different problems in immunosuppression as kind of a next step, and think about how we can work, again, towards more evidence-based foundation for the way that we do immunosuppression after pediatric liver transplant.